Now let's look at the stage which is called as the pro-metaphase, as to what happens during pro-metaphase. Pro-metaphase is the beginning of the metaphase, and kinetechores, which were the proteins that were present, that was holding the two piston chromatids together, they start appearing. Kinetechores have a very important role, because eventually we will find that kinetechores actually stick to the microtubules, which forms the spindles. So kinetechores will start appearing. The spindles is a region which is composed of something we learned a long time back. We heard of a term which is called as a microtubule. Remember when we were talking about the cell, we said they're microtubules, and I gave you examples of actin and filament, and there were three different kinds of cytoskeleton that's present in the cell. Microtubules are mainly proteins that will give a little structure to form what are called as a spindle. The spindle is composed of more like a fibrous structure, and they sort of overlap many strands. The chromatids are now adhering to these spindle fibers with their kinetechores. Now, what happens during metaphase? Metaphase is probably the easiest to recognize, because during metaphase, what's most important is the chromosomal alignment is what is called as equatorial. If you have to recognize a cell, whether it's in metaphase or not, it's probably the easiest to recognize, is because during metaphase, you see all of these chromatids are aligned as equatorial plate in the center of the spindles, and they are attached by their kinetechores to the spindle fibers. And at this stage, also notice that the centromeres are exactly to the opposite end, and the spindle is completely formed. Metaphase is followed by the next stage, which is called as the anaphase. In the anaphase, the two sister chromatids are pulled apart. So the kinetechores splits out, and the two chromatids are pulling apart, and they're pulling towards their respective centromeres or respective poles. Now, as soon as they start splitting, they are now unduplicated. Remember, when they were as chromatids, they were duplicated strands. Now this is the first stage that we see that they're actually separating out, and they're moving towards their respective fold. And the way they're moving, you can see that eventually each pole will have the same number of chromosomes as they have been duplicated, and eventually they will form the two double helix strands. So notice there were 4 of them in the beginning, and at the end we still have the same number, which is 4. Now, as soon as the chromatids have separated and they have gone to each pole, a stage called as telophase is going to follow. Telophase is the reverse of actually prophase, which means that remember, we said earlier the nuclear membrane had disappeared. It starts appearing back. The nuclear membrane, sorry, sometimes it's hard to write with this pen. Try to write a little better. Nuclear membrane reappears. Nucleolus, which also disappeared, is going to start appearing. And another thing that starts happening is slowly that the chromosomes are getting thin again. So the thick region is getting less, and they start pulling out and lengthening, because eventually the chromosomes will become the chromatin or the indistinct chromatin as they were, or the invisible chromatin as they were. So almost this is telophase is the reverse of prophase, and after telophase comes the second part of mitosis. Now, in the second part of mitosis, remember, I said earlier, if it was a prokaryotic cell, it divides, separates out by binary fission. However, most eukaryotic cells will undergo cytokinesis. Cyto means cytoplasm, and kinesis is division. So it literally means division of the cytoplasm. The division of the cytoplasm is slightly different in animal cell versus the plant cells. In animal cell, it's more like the contractile ring, or a furrow is formed between the two cells, and it starts like each cell is forming a furrow from both sides, and it's pinching, and it's eventually separating out into cells. However, in plant cell, a cell plate is formed. So I'm going to make you another diagram over here, which is a little simpler to understand. A cell plate is actually a cell that will form cell walls on both sides, which will eventually separate out. So there's a cell plate growing on this end and another plate growing on this end, and eventually a cell plate is formed and it divides the cell. So cytokinesis is a process by which the cytoplasm is separated out, and along with the cytoplasm, it has separated the organelles that are present within the cytoplasmic region, and the cell is now very similar to its appearance as it was during the previous interphase. And at the end of cell cytokinesis, a cell cycle has permitted a formation of identical new cell. And this stage is almost the end now. I'm going to try to go to the next slide. Yeah, this is the last slide, because and then I'm going to divide my lecture for the cancer in the next little break. So what I want to do is I want to go back and review the image that I had over here, which was this image. Actually, was it this image? No, this was. Yeah, this is this image. So keep in mind we had prophase, in which the cell was starting to form the spindle. The centromeres are pulling around on both ends. The nuclear plasm is disappearing. The nuclear membrane is disappearing. The chromatin is becoming thick, and eventually, I'm going to go through slides just quickly over here so that you don't have to go back and review each. Okay. So here it is, pro-metaphase. Quickly, notice how the chromosome are aligning on the spindle, but they're not as equatorial as they are. And the equatorial stage is hence called as metaphase. Metaphase is then followed by the stage which is anaphase, in which the chromatids are pulled apart, and then eventually anaphase is followed by telophase, in which a furrow develops an animal cell, and nuclear membrane is appearing. The chromatids is becoming thinner, and nucleolus also starts appearing. And that concludes with the cytokinesis. So the stages, each stage of the cell division is important. Each stage you should be able to recognize from an image as to which state it belongs to. There are some key features in there which helps you recognize as to which stage the chromosome is in. I'll probably have you draw some of these diagrams in class, and we will review some. And make sure you watch the animation on this section, because it's really very good. And then this concludes this section of mitosis, and I'll go ahead and end the chapter and the cell division of the cancer in the next part.